The first cases of SARS-CoV-2 were reported in December 2019 in the city of Wuhan, China. From then, the virus spread at alarmingly rapid rates to other parts of the country. In January 2020, WHO described the virus as the cause of a new disease, COVID-19, which was officially characterized as a pandemic in March 2020. As of today, the number of confirmed cases around the world is close to 6 million, with deaths over 370,000. SARS-CoV-2 belongs to the coronavirus genre of the Coronaviridae family, and it's characterized by having an envelope and a positive single-strand RNA genome. Despite its worldwide spread, the virus' epidemiological and clinical behaviors have not been established. How the COVID-19 affects children is one of the most enigmatic questions. As some observational studies show, its clinical expression in pediatric patients is not quite similar to the adult one.
Therefore, the objective of our project is to answer the following question. Why do children seem to present less severe manifestations of the COVID-19 disease? Well, there are many existing differences between adults and children that could justify their difference in their response to SARS-CoV-2 infection. After a thorough revision of the existing literature, considering both the transformations of the human body with AIDS as well as the virus characteristics, we have decided to center our investigation around three particular aspects: the influence of ACE2 receptor levels, the immune response, and the microbiota composition. This way, we're able to formulate three particular hypotheses that on one way are backed up by scientific literature, and on the other hand, could open new lines of treatments for those that are affected by the COVID-19 disease.
To test this hypothesis, we would conduct an observational retrospective study with an expected duration of 2 years. We would recruit a total of 1,000 subjects, both healthy and COVID-19 positive children from 2 to 14 years old, and adults from 15 to 80. COVID-19 positive diagnosis would be confirmed by a reverse transcription PCR. Sample analysis would be performed in the Barcelona Biomedical Research Park and the Genomics Regulation Center, who have generally provided their facilities.
ACE2 is a cell receptor present in multiple cell types that acts as SARS-CoV-2 gateway to the host. Moreover, it has a physiological role controlling blood pressure by converting angiotensin II into angiotensin 1-7. There is inconsistent literature on whether ACE2 acts as a protective or a risk factor when it comes to facing the virus. Besides, an age-dependent ACE2 expression has been reported in some animal models such as sheep and mice, although findings are also contradictory. While in mice ACE2 decreases with age, in sheep it increases. This contradiction makes us want to study ACE2 expression in humans, to see if an age-dependent expression also exists, and if so, determine its tendency. Our hypothesis is that ACE2 receptor levels are higher in children than in adults, offering protection, explaining why children present less severe symptomatology. And how are we going to prove this hypothesis? First, we would measure them indirectly through quantification of angiotensin II levels in serum by doing an ELISA. Secondly, we would directly study the receptor's expression by performing RNA sequencing. This technique would allow us to obtain and study the transcriptome of the tissues extracted.
As cross-reactivity between rubella IgGs and SARS-CoV-2 has been proven, in this study we would test whether other specific IgGs could also be inducing cross-immunity. We would focus on the two other viruses present in the MMR vaccine, the measles virus and the mumps virus. Following blood extraction, we would use serum samples from a COVID-19 positive group of vaccinated children and children who are not vaccinated nor present immunization due to previous infections. COVID-19 negative patients would be our control group. By doing two separate ELISA assays, we would test specific antigens from both mumps virus and measles virus against COVID-19 positive and negative patient serums. Higher levels of IgG in COVID-19 positive patients than in negative controls would indicate that a secondary immune response is activated. We would then compare the symptoms of vaccinated children with those not immunized to determine whether this cross-immunity has an effect on the severity of the COVID-19 disease. It has been shown that exaggerated inflammatory response secondary to high levels of pro-inflammatory cytokines in adults causes most of the damage they suffer. This increase is directly associated with cytokine storms that can lead to acute respiratory distress syndrome. To test whether these cytokine levels are lower in COVID-19 positive children than adults, we would perform an ELISA assay with both children and adult serums. We would also seek for an association between high cytokine levels and a greater thromboembolic risk, demonstrating their role in the severity of the disease. As it has been suggested that TNF-alpha inhibitors can modulate the cytokine storm, medication against TNF-alpha would be introduced as a variable to see whether this results in milder symptoms and a lower risk of thromboembolism.
The microbiota is defined as all the microorganisms inhabiting a region of the body. Human microbiota changes with age. Its diversity increases as the person grows older, but the total number of microorganisms decays. Although nasopharynx and bronchus microbiota are not completely equivalent, both can be used to compare respiratory microbiota between subjects. The role of this microbiota could be important in the development of COVID-19, as dysbiosis in the respiratory microbiota has already been associated with several diseases. Moreover, presence of specific viruses or bacteria in the mucosa of children's airways could have an impact on SARS-CoV-2 growth due to bacteria-to-virus and virus-to-virus interactions, as well as their competition for the niche. Our hypothesis is to test if having a more abundant nasopharynx and bronchus microbiota, as well as a greater number of specific microorganisms, is a protective factor. In order to do so, we would compare the bacterial 16S or RNA coding gene of the microbiota obtained from the nasopharynx swabs of the subjects of each group. By proving that AC2 is a protective factor, we would establish the administration of AC2 as a promising treatment.
Establishing higher cytokine levels in adults would be useful to recommend the use of some treatment alternatives such as cytokine inhibitors. Specifically, proving that TNF-alpha has a protective role in the severity of the disease would allow for the use of the mentioned medication in patients with a higher risk of thromboembolism.
If antibodies against measles and mumps prove to be a protective factor as well, vaccinations against these two infectious diseases would be seen as a prevention treatment.
Finally, proving the protective effect of certain microorganisms or a more densely populated microbiota would promote the use of probiotics in risk patients. Moreover, it would disencourage the use of treatments that significantly decrease the population of the microbiota.
In conclusion, although the ideal way of fighting the disease involves a specific vaccine, in the meantime, we hope that our project will be a helpful guide in terms of prevention and treatment for subjects and therefore be prepared for future possible scenarios involving the COVID-19 disease.